The ocean under moonlight is never silent. It hums with unseen currents, whispers of plankton blooms, and the restless bodies of predators patrolling between darkness and silver reflection. Along the rim of a Pacific atoll, where a narrow reef pass channels the tide like a funnel, a tiger shark drifts into view. The faint glow of the moon above filters into pale stripes across her back, painting her body with ghostly bands that echo the forest predator she was named for. She is not cruising idly. Her path appears chaotic, jagged, an angular dance across the current, but there is method hidden in the erratic geometry. She is following a plume, a ribbon of information carried invisibly through the water. The ocean is a library written in chemical ink. For a shark, the nose is the first interpreter. The tiger shark swings her broad snout side to side, sampling the water, slicing the current into alternating halves like a metronome. The oscillation is not random. It is a stereo test, a comparison of left and right, detecting which nostril receives the stronger signal. This is called stereo olfaction casting. In water thick with overlapping scents, the tactic allows her to home in on a trail that might be no more concentrated than a drop of blood diluted into billions of molecules. For humans, this dilution would be nothing. For the shark, it is as sharp as a flare across the sky. She turns left, then veers back right, adjusting her angle with every flick of her head. Slowly, like a needle stitching fabric, she weaves herself toward the origin of the plume. Research on tiger sharks has revealed that they do not simply wander aimlessly across their habitats. Field observations, combined with hydrodynamic modeling, show how they tack like sailors against invisible ribbons of odor drifting in and out of reef passes. These plumes can carry information for kilometers, bending with currents, splitting around coral heads, rising or sinking with density gradients. At night, when vision loses dominance, this chemical topography becomes the primary map. Every reef pass is a portal through which scent escapes, carrying the biological stories of spawning fish, injured turtles, or rotting whale fall remains lodged in a channel. The shark is not merely hunting, she is reading. Her nose is tuned to the physics of dispersal, and the jagged pattern of her swim is the translation of turbulence into intent. Yet the nose alone is not enough. As the plume narrows, the shark moves into final approach. Here, the ampullae of Lorenzini take over, the jelly-filled electroreceptors scattered across her snout like invisible freckles. At the last meter, where chemical concentration fades into ambiguity, electrical cues from muscle contractions and heartbeat finish the story. The prey may be buried in sand, hidden among reef ledges, or stunned in exhaustion. To the shark, the faint voltage field betrays it. In this final second, her navigation shifts from chemistry to physics, from diffuse plumes to localized signals. This two-tiered sensory strategy, long-range chemical casting, short-range electroreception, explains why tiger sharks succeed as generalist hunters across oceans. They move seamlessly from one information system to another, always adjusting the lens of perception. But the tiger shark's travels extend far beyond nightly foraging loops. These predators are not confined to a single reef or bay. Satellite tracking has revealed migrations stretching hundreds, even thousands of kilometers, linking distant islands and open ocean feeding grounds. What guides them across such featureless expanses, where neither odor nor landmarks can persist? Increasing evidence suggests that tiger sharks, like sea turtles and salmon, possess a magnetic map. Iron particles embedded in their tissues, combined with neural sensitivity, allow them to perceive Earth's geomagnetic field as a navigational grid. In controlled studies, sharks exposed to altered magnetic conditions shifted their swimming orientation, as though their internal compass had been nudged. The implication is profound. Tiger sharks may use geomagnetic signatures to recognize specific locations, homing toward reef passes with the precision of a returning bird. This magnetic sense gives meaning to their appearance at predictable channels year after year. A reef pass that funnels fish spawning aggregations each spring becomes not just a seasonal buffet, but a memorized coordinate in the shark's mental atlas. Long migrations through open water are not aimless drifts, but deliberate lines between magnetic waypoints. The tiger shark embodies a fusion of navigational strategies, plumes and voltages for the small scale, geomagnetism for the large. In both, physics becomes the roadmap of survival. Yet the modern ocean has changed. Human activity lays down its own artificial trails, and tiger sharks follow them with equal dedication. Harbors where fishing boats unload their catch, or channels where discarded fish waste drifts out on the tide, create powerful scent corridors. Chum slicks, offal discharge, and discarded carcasses produce plumes far stronger than natural prey odors. Sharks learn these artificial cues and adapt their patrols accordingly, turning human garbage into reliable feeding stations. What was once a migratory route between reef passes may now include a detour into harbors, docks, or marinas. These altered behaviors come with risk. A shark conditioned to follow human plumes is more likely to encounter fishing gear, boat traffic, and the lethal edge of human intolerance. Necropsies of stranded tiger sharks tell this story in brutal detail. Scientists have documented stomachs filled not only with the expected remains of turtles, fish, and seabirds, but also hooks, fishing line, and plastic debris. One shark carried the remnants of a license plate, another the rubber casing of a boot. These are not myths, they are real forensic records. The tiger shark's reputation as an indiscriminate eater reflects its ecological role as a scavenger, willing to sample anything that carries odor or signal. But the human footprint has tipped the balance. A hook swallowed with fish waste becomes a hidden killer, embedding in the gut or jaw. Plastic fragments, mistaken for prey or carried in on carcasses, clog or puncture. In these post-mortem stomachs, the convergence of natural instinct and artificial environment is carved into tragedy. What emerges is a paradox. The tiger shark is both one of the most finely tuned navigators of the ocean and one of the most vulnerable to the distortions we introduce. Its body is a sensor array built across millions of years. Stereo smell for turbulence, ampullae for microvoltages, magnetoreception for planetary scale. Yet those same abilities make it susceptible to following the wrong trails, mistaking garbage for food, or turning artificial harbors into permanent patrols. In this sense, tiger sharks are not random marauders. They are interpreters of physics, mapping the world through plumes, voltages, and fields. Where we create new roads, through waste, chum, or industrial discharge, they follow them with equal faith. Under the moonlit reef pass, the shark finally resolves her plume. The zigzag tightens into a straight line. She angles upward, jaws parting just enough for a flicker of serrated teeth to catch silver in the lunar glow. A school of jacks, restless and twitching, had spilled a wounded individual into the current. The chemical ribbon from its injury wound had spread through the channel, unraveling like a flag. The shark closes in, the ampullae registering the final flutter of muscle. A sudden surge, a single snap, and the trail is resolved into flesh. Silence returns, but the story remains, a predator that stitched physics into dinner. The scene fades, but the lesson is clear. The tiger shark is not a brute force machine of appetite. It is a navigator in an invisible world, moving between scales of perception that humans can barely imagine. For every apparent randomness in its path, there is an underlying order. The plumes of chemical dispersal, the whisper of bioelectricity, the steady pulse of the planet's magnetic field. And in those same systems, our footprint is etched, the waste that creates false roads, the plastic that turns scavenging into poisoning, the hooks that mimic prey until it is too late. The tiger shark teaches us that to understand predators, we must first understand the physics they inhabit. They do not swim blindly through the ocean. They follow the maps that evolution gave them, and now increasingly the maps we have imposed. Beneath the swell of the reef slope, where sunbeams fracture into shafts of green and blue, there is a den that glows faintly in the half-dark. The glow is not from light itself, but from reflection. Thousands of pearl-like eggs shimmer in tight clusters, dangling in strings from the rock ceiling. Each capsule contains the outline of a new life, embryos stirring as their chromatophores begin to form faint stripes of color. Guarding them is their mother, an octopus who has not eaten in weeks. She has sealed herself within this chamber, ignoring the scuttling of crabs at the entrance and the drifting silhouettes of passing fish. Her hunger is gone, not because there is no food, but because something deep inside her body has switched it off forever. The optic gland, a small mass of tissue near her eyes, is the architect of this transformation. In the life of an octopus, the optic gland functions almost like a master hormonal switch. When reproduction begins, this gland alters its secretions, flooding the body with chemical signals that suppress appetite, heighten maternal care, and prepare the mother for a single, irreversible sacrifice. Experiments have shown that removing the optic gland can extend an octopus's life dramatically. Mothers deprived of this gland's command will resume hunting and survive months beyond their natural span. But intact, the gland enforces the rule of nature. Once eggs are laid, the mother must starve so her energy and attention never waver from her brood. This biological surrender is astonishing in its extremity. For months, the female octopus will remain in her den, blowing jets of water over her eggs to keep them oxygenated, plucking away bits of sediment and fungi, defending them against intruders with sudden bursts of color and sharp beak strikes. Even when a crab stumbles brazenly past her arms, brushing the very suckers that could grasp it, she does not strike. The gland has reprogrammed her priorities, shifting her from hunter to guardian. Her own tissues waste away as she invests everything into the next generation. By the time her eggs hatch and drift into the open sea as planktonic larvae, the mother will be too weak to continue. She will collapse at the mouth of her den, her sacrifice complete. To understand the magnitude of this transformation, one must first remember what the octopus is at her prime. In the open reef, she is not just a hunter, she is a living net with 8 muscular arms arranged like a parachute. When prey wanders too close, she launches in a coordinated pounce, spreading her webbed arms wide so that her body becomes a canopy. The prey, whether it is a fish, shrimp, or crab, finds itself suddenly trapped within a tent of living flesh. Each arm is covered with suckers that not only grip but taste, sampling the texture and chemical signature of what they touch. The octopus does not see her prey only with her eyes. She tastes it through her arms, feeling not just shape but flavor. Her skin becomes an extension of her tongue, and her arms a sensory orchestra that gathers information with each curling movement. This chemotactile exploration is not limited to food. Every rock, shell, and coral fragment touched by the octopus is read like a script of the reef. With this information, she decides whether a stone can be used as a barricade, a shell repurposed as a door, or a crevice chosen as a safe den. Observations across the world's reefs have revealed octopuses dragging coconut halves across the seafloor to use as shields, stacking rocks at the mouths of their dens to keep predators at bay, and even carrying shells under their arms as portable armor. Each act is not instinct alone, but an improvisation, an application of intelligence to the demands of the moment. This tool use has provoked debates far beyond the reef, raising ethical questions about whether octopuses should be treated as mere commodities in the rise of octopus farming. If an animal can manipulate tools, recognize itself in tasks, and express complex behaviors, is it just seafood, or is it something more? Perhaps the most haunting glimpse of the octopus's mind comes during sleep. In recent years, scientists filming octopuses at rest have documented episodes that resemble rapid eye movement sleep in mammals. The animal's skin flashes through waves of color, chromatophores expanding and contracting in patterns that mimic hunting camouflage, defensive displays, or mating signals. Some researchers hypothesize that these are dreamlike states, rehearsals of memories encoded in the octopus's neural networks. Given that an octopus's nervous system is distributed, with more than half of its neurons residing in the arms rather than the central brain, such states may involve the whole body remembering together. The spectacle is eerie, a sleeping octopus pulsing through kaleidoscopic disguises, as if reliving hunts or encounters within its dreams. All of this, hunting mastery, tactile tasting, problem solving, even possible dreaming, collapses into silence once motherhood begins. The optic gland does not allow distractions. By suppressing feeding and redirecting neural priorities, it ensures that the mother becomes a machine of protection. Her death is the final investment. In evolutionary terms, this strategy may seem brutal, but it makes sense for a creature with such short lives and vulnerable offspring. Octopuses rarely live beyond a few years. Their larvae, floating as microscopic specks in the plankton, face extraordinary mortality. By devoting every last reserve to guarding eggs until the moment of hatching, the mother maximizes the survival odds of at least some of her offspring. There is a paradox here, one that has captivated biologists. The octopus embodies both extreme intelligence and extreme mortality. Unlike mammals, where intelligence is supported by long lifespans and extended parental care, the octopus condenses its brilliance into a brief, blazing span. It learns, it solves puzzles, it improvises tools, and then it dies before the next generation begins. Its intelligence is therefore built not on continuity but on sacrifice. The mother ensures her offspring by erasing herself. The ethical debate over octopus farming arises directly from this tension. As demand for octopus meat grows globally, companies have proposed captive rearing programs. But critics argue that farming an animal capable of tool use, memory, and problem-solving is fundamentally different from farming fish or shellfish. Octopuses resist captivity. Their intelligence makes them escape artists, capable of unscrewing jars, dismantling plumbing, or squeezing through tiny gaps. Farming them not only challenges logistics, but raises moral questions about confining such sentient creatures to sterile tanks. When paired with the reality of the mother's sacrifice in the wild, the debate deepens. Are we reducing an animal capable of such profound maternal devotion and potential dreaming states into nothing more than mass-produced protein? Back in the reef den, the mother's vigil continues. The crab that had walked by remains alive, untouched, scuttling away into shadows. The eggs glow faintly with life, each one pulsing as cilia stir within, moving yolk to mouth. The mother hovers close, her siphon gently directing streams of water across the cluster, ensuring that oxygen flows steadily. Her eyes, large and unblinking, reflect the dim light like glass orbs. She does not blink, does not eat, does not sleep in the way she once did. She simply waits, sustained by a biological imperative written into her optic gland. Weeks pass. Her skin begins to dull, her arms weaken, her mantle shrinks as muscle wastes away. The eggs hatch in waves, releasing clouds of planktonic hatchlings that scatter into the water column like sparks from a flame. They rise toward the open sea, where currents will scatter them far and wide. Each is barely larger than a grain of rice, yet each carries the genetic memory of camouflage, problem-solving, and the potential to repeat the cycle. Behind them, the mother collapses at the entrance of her den. She does not leave a body for scavengers. Her tissues will dissolve and nourish the reef she once hunted. In this moment, the octopus embodies a contradiction almost impossible for humans to grasp. A creature of immense intelligence that gives up its life not because of weakness, but because its biology demands it. Extreme intelligence balanced by extreme sacrifice. The mother who stops eating reminds us that evolution does not design for sentiment. It designs for survival of the lineage, even if it demands the obliteration of the individual. And in that obliteration, the reef gains a legacy, not in a single body, but in thousands of drifting larvae, each with 8 arms, each ready to taste the world through suckers, each carrying the possibility of brilliance into the future. In the dim shallows of a coral reef, where sand patches meet coral rubble, a predator waits inside a burrow no wider than a thumb. Its body is small compared to the sharks, turtles, and octopuses of the reef, yet its reputation reverberates across scientific literature and fisher tales alike. This is the mantis shrimp, an animal no longer than a human hand, yet armed with one of the most devastating weapons in the animal kingdom. The entrance of the burrow stirs, and the creature emerges, its raptorial club unfolding in slow, deliberate arcs. Within moments, that club will accelerate faster than the blink of an eye, generating a shockwave so powerful that it not only stuns prey, but briefly ignites light in the form of a cavitation flash. The mechanics of this strike defy intuitive understanding. When the mantis shrimp swings its club, the acceleration exceeds thousands of times the force of gravity. The speed is so extreme that the surrounding water cannot follow, and a void is momentarily created. This is cavitation, the sudden formation of vapor bubbles when local pressure drops below the vapor pressure of water. But the violence comes not from the bubble itself, but from what happens next. Within fractions of a millisecond, the bubble collapses, imploding with a shockwave that can exceed several hundred atmospheres of pressure in localized bursts. The result is 2 strikes for the price of 1. The physical club impact followed immediately by a secondary strike from collapsing cavitation. This phenomenon is not just physics trivia, it is survival. The mantis shrimp relies on this dual mechanism to shatter the shells of crabs, snails and mollusks that would otherwise resist predators. The club itself, shaped like a hammer, smashes with direct force. But if the prey resists, the cavitation bubble ensures that even the strongest armor fails. Sometimes, even if the club misses direct contact, the shockwave alone is enough to stun or kill. For an animal weighing only a fraction of a kilogram, this amplification of power allows access to prey usually reserved for much larger predators. In effect, the mantis shrimp cheats the size hierarchy, using physics to level the field. Yet the club that delivers this force is not fragile. Inside, its microstructure reveals a masterpiece of evolutionary engineering. Layers of chitin and mineral fibers are arranged in a helicoidal pattern, twisting like a spiral staircase. This design is called a Bulligand structure, and it allows the club to dissipate immense energy across multiple layers. Rather than cracking under repeated blows, the helicoidal arrangement forces fractures to twist, turn, and branch until they lose energy before spreading. Engineers have studied this design intensely, replicating it in advanced ceramics, protective gear, and aerospace composites. What nature built for smashing shells now inspires human armor against bullets and impact. A mantis shrimp club can withstand thousands of high-speed strikes without shattering, a feat no simple material could achieve alone. In the shallow reef, this weapon reshapes the ecosystem. Snails that would otherwise dominate substrate space are kept in check. Crabs risk becoming meals despite their exoskeletal defenses. Even small fish darting too close to a hunting mantis shrimp can be stunned by shockwaves. The predator thus occupies a unique niche, not the largest hunter, but one that influences shell availability, prey populations, and competitive balance among invertebrates. A single burrow can become an ecological pressure point, radiating influence through the reef as shells are broken, redistributed, or vacated. But the mantis shrimp's fame is not confined to its punch. Equally extraordinary are its eyes, complex, protruding structures that break every rule of conventional vision. While humans perceive light along 3 channels of color, red, green, and blue, the mantis shrimp perceives a spectrum divided into up to 16 channels. This includes the ultraviolet range invisible to us, and more remarkably, polarization. Polarized light is structured light, waves vibrating in a particular orientation. To most animals, this dimension of vision is absent. But to a mantis shrimp, polarization is a rich language layered atop the colors of the reef. In practical terms, polarization vision allows mantis shrimp to see contrasts invisible to fish and even to humans armed with cameras. The sheen of a fish scale, the reflective glare of a shell surface, or the orientation of light scattering off sand becomes coded information. Mantis shrimp use this to detect prey, otherwise camouflaged, to recognize rivals, and to communicate with each other. Some species bear specialized patches on their bodies that reflect polarized light, creating signals hidden from the wider reef community. To predators and competitors without polarization sensitivity, these patches mean nothing. To mantis shrimp, they are neon signs of identity, threat, or courtship. The evolutionary implications of this are immense. In a crowded reef where visibility is both vital and dangerous, mantis shrimp have evolved a private channel of communication, an encrypted signal that only those with the right visual keys can read. This allows them to engage in aggressive displays, territorial warnings, or mating rituals without drawing the attention of predators. For the human observer, polarization patterns can be revealed only with specialized filters, and even then, they lack the clarity with which a mantis shrimp perceives them. To us, the reef may shimmer, a tire. To the mantis shrimp, it speaks in coded bars and flashes of polarized light. Beyond the sheer spectacle, this sensitivity has practical applications that ripple into technology. Biomedical imaging, cancer detection, and advanced underwater cameras have all drawn inspiration from mantis shrimp vision. By mimicking their ability to detect subtle polarization differences, scientists have developed new ways of seeing tissue structures or enhancing contrast in turbid environments. Once again, the reef predator that smashes shells becomes an unintentional teacher to human innovation. The combination of shockwave strikes and hyperspectral eyes creates an animal both brutal and refined. It can kill with physics, yet it can perceive with nuance unmatched by nearly any other animal. The irony is that such sophistication arises in an animal with a nervous system far simpler than ours. Intelligence, it reminds us, is not a linear scale but a spectrum of adaptations. For the mantis shrimp, intelligence lies in how its sensory world is tuned to tasks: smash hard, see differently, communicate secretly. Evolution has given it exactly what it needs, not more, not less. In the reef ecosystem, the presence of mantis shrimp reshapes community dynamics in subtle ways. Empty shells created by their strikes are often colonized by hermit crabs, changing the distribution of shelter. Their polarization signals carve out social territories invisible to others, reducing unnecessary conflict. And their predatory focus on armored invertebrates keeps those populations from monopolizing benthic space. Like the tiger shark and octopus, the mantis shrimp reveals that dominance in nature is not always about size. Sometimes it is about exploiting the invisible: pressure waves, polarized light, or hidden structural designs. When filmed at high speed, the strike becomes even more surreal. The club arcs downward, a bubble blooms from nothing, collapses with a flash of light, and the prey's shell fragments in an instant. The mantis shrimp retreats, clutching its meal. For an observer, the violence happens too quickly to register with the naked eye. Only slowed footage reveals the choreography of forces at work: impact plus cavitation, a duet of destruction. The broader truth is this: mantis shrimp do not simply punch. They weaponize physics. They do not simply see. They unravel a dimension of light hidden to nearly all others. Each burrow, each strike, each flash of polarization is a reminder that ecosystems are shaped not only by visible interactions, but by invisible forces: pressure, light, structure, woven into biology. For the mantis shrimp, invisible pressure changes do the real damage, both in combat and in communication. It is a predator that lives not just in the reef, but in the realms of physics and optics, carving a world defined as much by unseen forces as by visible ones. The horizon is broken by nothing but water, a heaving plain of cobalt stretching into infinity. A storm has just passed, leaving the surface stippled with gentle ripples and a faint rainbow arcing low against the clouds. At the calm heart of this vast openness, a strange moment unfolds. A sea snake, banded in stark black and white rings, wriggles its way to the surface. It does not come to breathe alone, though air is its constant need. Instead, it begins to sip, mouth opening carefully to the thin film that floats above the salt. This is a rain lens, a shallow sheet of fresh water formed when rain pours onto the ocean, its lower density leaving it suspended for hours before mixing into the brine. To most creatures of the sea, the rain lens is meaningless. To the sea snake, it is life itself. Despite a body tuned for the ocean, it is still a reptile, and like all reptiles, it requires fresh water to drink. Out here, hundreds of kilometers from land, the only relief comes when the sky itself delivers it. This small scene reveals the paradox of sea snakes. They are reptiles by heritage, descended from terrestrial snakes that slithered through coastal mangroves and estuaries. Evolution has since sculpted them into some of the most specialized marine vertebrates on Earth, yet vestiges of their ancestry remain etched into their physiology. The need for fresh water is one of those echoes. Unlike fish, they cannot desalinate seawater effectively. Instead, they time their hydration with storms, rising to the surface to drink from the temporary freshwater sheets that form like transparent mirrors. Studies of their stomach contents and field observations confirm that after heavy rainfall, sea snakes often gather at the surface, their tongues flicking against the thin lens in synchronized desperation. For them, rain is not just a weather event, it is a hydration ritual dictated by physics. But once submerged, their adaptations tell a very different story. A sea snake's body is a narrow cylinder wrapped in scales that reduce drag, while the tail has flattened into a paddle-like fin that sculls with each undulation. Along the head, the nostrils are equipped with valves that clamp shut when the snake dives, preventing seawater from flooding its lungs. The lung itself is extraordinary, extended along nearly the entire length of the body. In terrestrial snakes, this elongated lung evolved to maximize space within a narrow frame. In sea snakes, the same structure doubles as a buoyancy control organ. By adjusting the volume of air, the snake can fine-tune its position in the water column, rising effortlessly to the surface or hovering mid-depth with minimal energy expenditure. It is both a scuba tank and a ballast system, a perfect repurposing of ancestral anatomy. The efficiency of this system is further enhanced by their ability to extract oxygen not just through lungs, but through their very skin. While sea snakes are reptiles bound to lungs for gas exchange, their skin has evolved to permit cutaneous respiration. In some species, up to a quarter of the oxygen budget can be met through diffusion across the skin surface. This allows dives to extend for half an hour or longer, remarkable for an animal whose body plan was never originally meant for submersion. It is a compromise, skin thin enough to permit gas exchange, yet still covered in scales tough enough to resist the abrasion of reef and sand. In effect, their skin has become a partial gill, an evolutionary innovation that bridges two worlds without abandoning either. These layers of adaptation are not confined to breathing and buoyancy. In recent years, researchers discovered that certain species of sea snake possess light-sensitive cells in the skin of their tails. At first, the finding was puzzling. Why would a snake need to see with its tail? The answer likely lies in predation and concealment. Sea snakes often retreat into reef crevices, wedging themselves into cracks with only their heads exposed. The tail, left protruding, becomes a liability, brightly patterned and twitching with movement. Light-sensitive skin may allow the snake to detect when its tail is exposed to open water or sunlight, prompting it to tuck the appendage deeper into the shadows. If true, this adaptation represents a form of distributed vision, skin acting as an extra set of primitive eyes, specialized not for detail, but for survival. On the hunt, sea snakes become active weavers of the reef fabric. They probe coral heads with slender bodies, tongues flicking to read the chemical environment, jaws ready to snap around small fish, and eels hiding in the labyrinth. Their fangs deliver a venom among the most potent of any snake, designed not for defense, but for quick immobilization of slippery prey. The bite injects neurotoxins that paralyze muscle, halting escape within seconds. Yet despite this arsenal, humans rarely suffer bites from wild encounters. Most incidents occur not when divers cross paths with snakes, but when fishing trawlers haul nets from the deep. Sea snakes, tangled in mesh and dragged to the surface, thrash in panic. Fishermen reaching in to clear the nets are at risk, and many envenomations occur here, in the violent meeting of industrial harvest and reptilian adaptation. Bycatch mortality is, in fact, one of the greatest threats to sea snakes today. Their habitats overlap with some of the most intensively fished regions of the Indo-Pacific. The snakes drown in nets, crushed under haul, or killed out of fear by those who encounter them. Ironically, the very biology that makes them so distinctive, the long lung, the reliance on fresh water, the narrow ecological window of shallow coastal seas, renders them vulnerable to collapse when human industry intensifies. In some regions, local populations have already declined sharply, their absence rippling outward through reef systems where they once played important predatory roles. Their ecological function is subtle but significant. By preying on small reef fishes and eels, sea snakes regulate mid-level predator populations. This, in turn, influences herbivore abundance and algal growth, cascading into reef health itself. They are not top predators like sharks, but their presence shapes balance in quieter ways. A healthy reef often supports multiple species of sea snake, each with slightly different hunting preferences and microhabitats, from sandy bottoms to branching coral forests. Remove them and the dynamics shift. Small fish boom, algae grows unchecked, and the reef begins to tilt out of equilibrium. There is a certain irony in how sea snakes are perceived. To most, they are symbols of danger, coiling silhouettes gliding silently through tropical waters. But to the ecosystem, they are regulators, opportunistic yet precise. And to scientists, they are marvels of evolutionary adaptation, land reptile tools, ocean-tuned in surprising ways. Their story is not just one of transition, but of compromise. They did not abandon their roots entirely. They still drink fresh water from rain lenses, still breathe with lungs, still lay eggs in certain amphibious lineages. Yet they layered upon this ancestry an arsenal of innovations, valved nostrils, cutaneous oxygen uptake, paddle tails, and even light-sensing skin. They are neither fish nor terrestrial snake, but something in between. Reptiles rewritten for a liquid world. As the rainbow fades and the rain lens begins to disperse, the sea snake takes one final sip before diving. Its body ripples in smooth undulations, tail sculling like a rudder. It weaves into coral shadows, hunting with chemical senses sharp as blades. Behind it, the ocean returns to stillness, the thin freshwater layer gone, mixed invisibly into salt. The moment is fleeting, but in its brevity lies the truth of the sea snake's existence, fragile in its reliance on rain, yet formidable in its mastery of the reef. A paradox suspended between storm and sea. The reef fades into darkness, and the open sand plain stretches ahead like a stage waiting for its performers. Moonlight filters weakly through the water, outlining pale ripples across the seabed. Then, from the edge of the rocks, a line of shadows emerges. First one lobster, then another, then another. Soon, dozens are marching in single file across the sand, antennae waving in synchronized rhythm, tails flicking dust with every step. To divers who have witnessed it, the scene looks like a nocturnal parade, sometimes stretching for hundreds of meters. Scientists call these formations lobster lines, and they represent one of the most remarkable coordinated migrations among marine invertebrates. These nighttime marches are not random gatherings. They are driven by seasonal changes, food availability, and breeding cycles. In tropical and subtropical regions, spiny lobsters often leave their reef shelters en masse to journey across sandy plains toward deeper waters. The line formation is more than a spectacle. It is a survival tactic. By moving in procession, lobsters reduce their chances of predation. The first individuals may face higher risk, but the collective movement creates a wall of antennae and armored carapaces that discourages would-be hunters. Group travel also helps individuals conserve energy, as turbulence created by the lobster ahead slightly eases the resistance for those following. Much like migrating geese flying in V formation, lobsters employ physics to their collective advantage. Navigation across these featureless expanses relies on tools far subtler than eyes or memory. Experiments have revealed that lobsters can detect and respond to Earth's magnetic field, using it as a kind of internal compass. In controlled tanks where magnetic fields were artificially shifted, lobsters oriented themselves as though preparing to march in the wrong geographical direction, faithfully following the altered cues. This magnetic sense allows them to cross vast sandy stretches without landmarks, finding new shelters or returning to known reefs even after displacement. In essence, their lines across the seabed are not random strolls but magnetic marches, invisible maps etched into their nervous systems. But lobsters are not only navigators. They are also communicators, though their methods differ radically from what humans might expect. When two lobsters meet, conflict often seems inevitable. Claws clash, antennae whip, and postures stiffen. Yet beneath the visible displays lies a chemical dialogue. Lobsters release urine from openings near the base of their antennae, spraying it directly into the face of rivals or potential mates. This urine carries chemical signatures of size, sex, reproductive condition, and recent victories. A dominant lobster's urine effectively tells a challenger, "I have already fought and won." In many cases, this prevents physical combat, saving both animals from injury. In courtship, the same mechanism transmits readiness, setting the stage for pair formation. This form of chemical communication is so effective that lobsters can remember their social standings, avoiding unnecessary battles with individuals they have already learned to recognize. Researchers studying captive populations found that once dominance hierarchies were established, chemical cues alone were enough to maintain order. In the reef, this prevents endless cycles of injury and wasteful energy. It is a subtle but critical layer of lobster society, a world where streams of urine carry messages more persuasive than claws. Their ecological role extends far beyond these interactions. Lobsters are omnivorous, scavenging carrion, consuming algae and preying upon sea urchins. In many kelp forest ecosystems, their appetite for urchins plays a keystone role. Left unchecked, urchins graze relentlessly on kelp holdfasts, stripping forests until only barren seascapes remain. Where lobsters thrive, urchin populations are kept under control, and kelp forests persist as vibrant habitats supporting fish, invertebrates, and marine mammals. Remove lobsters through overfishing or disease, and the ecosystem can collapse into urchin barrens. In this way, the nightly marches of lobsters ripple outward into entire marine landscapes, balancing herbivores and preserving underwater forests. Yet the story of lobsters is not only one of ecological guardianship. It is also a story of human exploitation and unintended consequences. Lobster fisheries are among the most lucrative in the world, supporting coastal economies across the Americas, Africa, and Asia. But the traps used to harvest them, wooden or metal pots baited with fish, pose a hidden danger when lost at sea. Storms, currents, and accidents can sever trap lines, leaving gear abandoned on the seafloor. These ghost pots continue to function as deadly snares, luring lobsters inside and preventing escape. Trapped individuals die, their decaying bodies attracting more lobsters, which in turn perish and attract yet more. The cycle repeats for years, creating self-baiting death chambers. Ghost fishing is not confined to lobsters alone. Crabs, fish, turtles, and even diving seabirds can be ensnared in abandoned gear. Studies estimate that in some heavily fished areas, thousands of lost traps may line the seabed, killing silently and without yield. For lobsters, this loss is twofold. Individuals are removed from the breeding pool, and the ecosystem suffers from the ripple effects of their absence. Fisheries themselves also lose potential harvest, a paradox where human industry undermines its own resource. Solutions exist, but their adoption is uneven. One of the simplest is the use of rot cords, biodegradable panels or twines attached to trap entrances. If a trap is lost, the cord decays within months, opening an escape hatch and preventing indefinite killing. The technology is cheap and straightforward, yet in many regions enforcement is lax or non-existent. Where regulations require rot cords, ghost fishing declines dramatically. Where they are absent, the seabed remains littered with silent killers. The difference lies not in technology, but in policy and compliance. Viewed from above, the contrast is striking. On one hand, a nocturnal march of lobsters across sand, guided by magnetism, policing their societies with chemical signals, protecting kelp forests through their appetites. On the other, the wreckage of our interventions, traps turned into tombs, lines entangling marine life, and populations diminished not by predators, but by persistent human neglect. Lobsters, in this sense, are both beneficiaries of evolutionary ingenuity and victims of human inertia. The image of a lobster line is almost ceremonial. Antennae extend like banners, claws shift rhythmically, carapaces glint under moonlight. It is a procession rooted in ancient instincts, a survival choreography that has persisted for millions of years. Yet the march now unfolds in a world layered with new hazards. Magnetic maps still guide them across the sand, but ghost gear lies in wait. Urine still settles disputes, but dwindling numbers reduce the frequency of such encounters. Their predation still spares kelp forests from urchin destruction, but only where populations remain healthy enough to exert influence. What emerges is a paradox that echoes through many marine species. Evolutionary brilliance, shaped in a natural world, collides with human negligence in the modern one. The lobster is not just a crustacean of culinary fame. It is a navigator of invisible magnetic fields, a communicator through chemical codes, a keystone predator holding ecosystems in balance. But it is also a casualty of ghost gear, caught in death traps designed by the very species that reveres it as a delicacy. As the night wanes, the lobster line reaches its destination, a deeper reef where crevices and ledges provide shelter. The parade dissolves, individuals fanning out into the landscape, each antenna sweeping, each carapace slotting into cracks. The sand plain lies quiet again, but the trace of the march lingers. These are not mindless crustaceans wandering blindly. They are participants in a ritual shaped by magnetism, chemistry, and ecological necessity. Whether those rituals endure depends not only on the resilience of lobsters themselves, but on the choices we make about the gear we leave behind. The takeaway is sharp. Lobsters are keystone night patrols, essential to the balance of reefs and kelp forests. Yet unless ghost pots are curbed, our tools will turn their own brilliance into slow, pointless mortality. For every lobster line marching across the sand, there is another trapped in silence, a casualty of a problem we already know how to solve. The question is whether we will. The ocean at dawn is an endless plain of silver, broken only by faint slicks that stretch across the horizon like ghostly roads. These slicks are convergence fronts, formed where currents, wind, and temperature gradients corral floating matter into ribbons. To the human eye, they appear as subtle sheens, glistening under sunlight. To a green sea turtle rising from below, they are highways of opportunity and danger. Drifting among these bands are swarms of jellyfish, their bells pulsing rhythmically, their tentacles dangling in the slow ballet of gelatinous life. For a turtle, this is a banquet. Yet woven into the same slick are plastic bags, film and fragments, artificial twins of the jellyfish, riding the same physics. A turtle ascends, bites into a jelly, swallows and turns for another. Just behind it, a translucent bag flickers in the current. This is the paradox of modern oceans. Convergence fronts concentrate food and pollutants alike. The very forces that create highways of energy, gathering plankton, jellies and small fish, also gather humanity's discards. Green turtles, with eyes adapted to detect the faint translucence of their prey, struggle to distinguish between drifting cnidarians and polyethylene sheets. Ingested plastics block intestines, puncture tissues, or simply accumulate until starvation follows. For an animal that has been navigating these slicks for tens of millions of years, the new hazard is an evolutionary ambush, one their senses never prepared them for. Yet to focus only on this tragedy is to miss the larger truth. Green sea turtles are among the most remarkable navigators and ecosystem engineers of the marine world. Their life story spans entire oceans, looping across hemispheres with precision still only partly understood. Hatchlings emerging from nests on tropical beaches imprint on the geomagnetic signature of their birthplace. Tiny variations in Earth's magnetic field, invisible to us, are encoded into their nervous system like a compass rose. As they drift into the open sea, riding currents across entire basins, these magnetic signatures remain stored. Decades later, as adults ready to breed, they return with astonishing accuracy to the very beach where their lives began. Experiments manipulating magnetic fields around young turtles confirm this. When exposed to magnetic coordinates from distant regions, hatchlings orient themselves in the directions consistent with those coordinates. The ocean is not blank space to them, it is a map inked in magnetism. Between hatching and return, green turtles traverse thousands of kilometers, grazing seagrass beds, resting in caves, and hunting gelatinous prey along convergence lines. Their ecological role as grazers is profound. By cropping seagrass, they stimulate regrowth, much as terrestrial grazers like bison shape prairies. Seagrass blades regrow denser and more productive after turtle grazing, creating habitats for fish, crustaceans, and invertebrates. More importantly, seagrass meadows act as carbon sinks, sequestering vast amounts of atmospheric carbon in sediments. In this way, turtle grazing indirectly boosts what scientists call blue carbon, the ocean's capacity to mitigate climate change. Remove turtles, and seagrass becomes overgrown, shaded, and less productive. Keep turtles and the meadows thrive, locking carbon beneath the seabed for centuries. A reptile that first crawled onto beaches over 100 million years ago now plays a quiet role in buffering the Anthropocene. Yet green turtles also carry shadows in their biology. In many populations, individuals develop fibropapillomatosis, a disease characterized by grotesque tumors sprouting across eyes, flippers, and shells. These tumors, often viral in origin, blind turtles, slow their swimming, and reduce their foraging efficiency. The condition has been linked to degraded water quality, nutrient runoff, and pollution, which may stress immune systems or fuel viral expression. In coastal lagoons with heavy agricultural discharge, fibropapillomatosis prevalence soars. In cleaner waters, it declines. Thus, the disease itself becomes a living barometer of ecosystem health, a sign that turtles are absorbing, within their very bodies, the imprint of human land use. At rest, the turtle presents another marvel. In coastal caves or under ledges, they wedge themselves into rocky alcoves, their bodies motionless, their heads tucked. During these sleep bouts, their heart rates drop to astonishing lows. Some individuals reduce beats to 1 every 9 minutes, sustaining oxygen conservation that allows hours of rest without surfacing. This bradycardia, far beyond what human physiology could tolerate, reflects the reptilian mastery of metabolic suppression. It is not only during dives but also during stillness that turtles display their evolutionary economy. Energy saved is energy banked for journeys across oceans. Ecologically, every stage of turtle life weaves into marine systems. Hatchlings leaving nests provide pulses of energy to predators like birds and crabs. Juveniles grazing seagrass-shaped plant growth. Adults consuming jellies regulate blooms that, left unchecked, could disrupt fish populations. Even turtle carcasses, when they occur, become massive nutrient subsidies, feeding sharks, crabs, and deep scavengers. They are not singular organisms, but moving nodes of connection, shuttling energy and nutrients across realms. And yet, everywhere their biology collides with ours. Plastic bags drift along the very slicks where jellies gather. Fishing gear ensnares them, drowning turtles that surface too late. Rising seas threaten nesting beaches, while artificial lighting disorients hatchlings away from the ocean and toward roads. Fibropapillomatosis expands in coastal waters influenced by our waste. The geomagnetic imprints that once guided turtles unerringly to their natal beaches now lead some to shores transformed into resorts, ports, or seawalls. Still, green turtles endure. Populations once nearly exterminated by hunting for meat and callipee, used in soups, have rebounded in some regions through strict protections and conservation programs. Nesting counts in parts of the Caribbean and Pacific show hopeful increases, though threats remain uneven. Their resilience is testimony not only to their adaptability, but to the power of targeted conservation. When beaches are protected, when bycatch is reduced through turtle excluder devices, when seagrass meadows are preserved, turtles return. Their life history is long, but so is their memory. They come back when given the chance. Back at the convergence front, the contrast sharpens. A turtle bites into a jelly, tentacles trailing from its beak, swallowing a meal rich in protein and water. It turns for another, gliding along the slick where food is dense. Yet the same hydrodynamic forces that delivered the jelly buffet have also delivered a translucent bag, fluttering like a mimic. The turtle closes in, mouth opening. The decision is blind, dictated by evolution's tuning of vision for gelatinous prey. Whether this bite nourishes or kills depends not on the turtle, but on us. The lesson is stark but layered. Green turtles are ancient navigators, reading magnetic maps across oceans, grazing seagrass to keep ecosystems healthy, and sleeping with heartbeats slowed to near stillness. They are part of the architecture of the sea, keystone players in the quiet maintenance of balance. Yet into their world we have poured our newest pollutant, plastic, which rides the same physics as their food. To them, a slick is still a highway of survival. To us, it has become a mirror of responsibility. At low tide, the reef is still, pools shimmering in the retreating light. Among the rocks, something stirs slowly, almost imperceptibly. 5 arms radiate outward like spokes of a wheel, each covered with rows of tiny tube feet that rise and fall in coordinated sequence. The creature advances in eerie silence, gripping with hundreds of microscopic suckers, its body a mosaic of calcified plates. It is a starfish, an animal that challenges every assumption about what a nervous system or a predator should look like. The starfish does not have a brain. Instead, it operates through a decentralized nerve net that spreads across its body. Each arm contains its own neural circuitry, linked by a central nerve ring that coordinates movement and response. This distributed system allows astonishing autonomy. If one arm is injured or severed, the others can continue moving, steering the body away from danger. Experiments show that individual arms can even initiate feeding behavior when separated, demonstrating that intelligence in starfish is not concentrated in a single organ but diffused throughout their tissues. To us, accustomed to the dominance of the brain, the idea feels alien. To the starfish, it is survival. At the tip of each arm lies an eye spot, a cluster of photoreceptive cells sensitive to light and dark. These crude eyes cannot resolve detail but provide spatial awareness, allowing the animal to distinguish shadowed crevices from open water. Combined across 5 arms, the starfish gains a panoramic awareness of its environment. It does not need sharp vision to hunt. It only needs to know where the reef offers shelter and where prey clings to rock. The real power of the starfish lies beneath its surface, in a hydraulic system unlike anything in vertebrates. Water enters through a sieve-like structure called the madreporite, circulating into radial canals that extend into each arm. From these canals, tube feet extend outward, each filled with pressurized water. By contracting tiny muscles, the starfish controls the pressure inside, extending or retracting each foot with precision. At the tip, a suction disc grips surfaces through adhesion, allowing the animal to cling to rocks or pry open shells. The physics is both elegant and powerful. Hydraulics create force without muscles, a biological engineering feat that enables slow but relentless movement. This system is on full display when a starfish attacks a mussel. Clambering onto the shell, it positions its arms around the edges, hundreds of tube feet anchoring simultaneously. Slowly, with imperceptible but inexhaustible strength, it pulls. The mussel resists, its adductor muscles clamping the shell shut. Minutes pass, then hours, but the starfish is patient. Tube feet contract in waves, distributing tension across the shell until the mussel's muscles fatigue. A sliver of space opens, no wider than a fingernail. The starfish responds with its most astonishing weapon, its stomach. From the center of its body, the starfish everts its cardiac stomach, turning it inside out through its mouth and inserting it into the gap. Enzymes flood into the mussel's shell, liquefying tissues. The starfish does not eat by chewing or tearing. It digests externally, sucking the slurry back into its body. The prey's shell never moves more than a few millimeters apart, yet its body is emptied from within. It is a gruesome efficiency, a form of predation that requires neither speed nor aggression, only patience and an alien physiology. The regenerative powers of starfish add another layer of resilience. If an arm is lost to a predator or accident, it can regrow. In some species, a single severed arm, if it retains part of the central nerve ring, can regenerate an entire new body. This capacity has fascinated scientists for decades, as the cellular mechanisms involved, stem cell activation, tissue patterning, and nerve regrowth, may hold clues for regenerative medicine. For the starfish, regeneration is simply another survival strategy. Vulnerable though it may appear, it carries within itself the blueprint to rebuild. Yet starfish are not passive residents of reefs. They are ecosystem engineers, shaping marine communities by regulating bivalve populations. In temperate waters, the keystone role of starfish was famously demonstrated in experiments where they were removed from rocky shores. Without starfish predation, mussels exploded in abundance, crowding out other species and collapsing biodiversity. With starfish present, balance was maintained, space freed for barnacles, algae, and fish. This single predator, moving at the pace of inches per minute, acts as a sledgehammer of ecological order. But starfish populations themselves are not immune to disruption. In recent years, sea star wasting disease has swept across North America's Pacific coast, devastating populations of multiple species. Affected individuals develop lesions, lose arms, and disintegrate into white goo within days. The precise cause remains contested. Viral agents, bacterial infections, and environmental stressors have all been implicated. Warmer waters may exacerbate the outbreak by weakening immune responses. Whatever the trigger, the collapse of starfish populations has unleashed cascading effects, with mussels and sea urchins surging unchecked, altering kelp forests and rocky shorelines. The absence of a soft-bodied predator becomes a hard blow to ecosystems. In other regions, the opposite problem occurs. Crown-of-thorns starfish, a venomous species covered in sharp spines, has proliferated to outbreak levels on coral reefs. Normally part of the reef community, these starfish feed on coral polyps, consuming living tissue and leaving behind skeletal skeletons. Under natural conditions, they are controlled by predators and limited food availability. But nutrient runoff from agriculture and coastal development fuels plankton blooms, providing abundant food for crown-of-thorns larvae. Populations swell and reefs are overwhelmed. Entire coral systems can be stripped bare in outbreaks, reducing structural complexity and biodiversity. Here, the starfish is not the maintainer of balance, but the architect of collapse. Both scenarios, wasting disease and crown-of-thorns outbreaks, demonstrate the double-edged nature of starfish influence. They are neither inherently destructive nor benevolent. Their role depends on context, on the delicate interplay of environment, predation, and nutrient flow. They are soft-bodied sledgehammers, capable of both building resilience and wrecking ecosystems. Even in their adhesives, starfish inspire science. The glue secreted by tube feet allows them to cling to rocks with remarkable strength, yet they can release instantly when they choose to move. This reversible adhesion, stick and unstick on command, has become a model for biomimetic research. Engineers study the molecular composition of starfish glue and anti-glue to design new medical adhesives, underwater sealants, and surgical tools. What evolved to grip barnacle-encrusted rocks now points toward solutions for human industry and healthcare. The starfish embodies the alien logic of the ocean. A nervous system without a brain orchestrates coordinated feeding and regeneration. A hydraulic network substitutes for muscles. A stomach that turns inside out accomplishes digestion beyond the body. Regeneration rewrites injury into renewal. And yet, despite this strangeness, their impact is immediate and real. They control prey, shape habitats, suffer mysterious diseases, and sometimes, under human influence, explode into destructive numbers. Back on the reef, the starfish retracts its stomach, leaving behind the empty shell of a mussel. Its tube feet march on, hundreds rising and falling in rhythm, gripping and releasing with perfect hydraulic precision. It has no brain, no central command, yet every movement is purposeful, every act integrated into a life history that binds it to the ecosystem. To us, the starfish is an emblem of simplicity. To the reef, it is a force of change. The reef slopes into deeper water, where currents rise against coral outcrops and sweep nutrient-rich flows upward. Here, at the meeting point of tide and rock, shadows gather. One by one, manta rays circle above the coral heads, their enormous wings carving arcs through the water. They seem like specters at first, gliding silently, but as the sun filters through the surface and strikes their backs, the patterns emerge, constellations of pale spots and inky rosettes unique to each individual, as distinct as fingerprints. One manta banks sharply and rolls, presenting its belly to a cloud of cleaner wrasses waiting among the coral. The fish dart forward, plucking parasites and dead skin, while the manta hovers almost motionless. It is not a chaotic feeding frenzy, but a ritual. There are rules here, unwritten but enforced. Distance maintained, turns taken, patience observed. For animals that gather by the dozen, etiquette governs survival. The manta ray embodies paradoxes. It is vast, its wingspan can stretch wider than a small car, yet it survives on the tiniest prey, plankton invisible from more than a meter away. It is a filter feeder like a whale shark, yet its brain is proportionally larger than that of almost any other fish. Studies of manta neuroanatomy reveal folds in the cerebral cortex more complex than expected, especially in the cerebellum, a region associated with motor coordination and learning. They are not mindless giants drifting with currents. They are thinkers, navigators, and social learners. Observations suggest they can recognize themselves in mirrors, an ability once thought limited to primates, dolphins, and elephants. They have been seen revisiting the same cleaning stations across years, adjusting approach depending on crowding, and even engaging in coordinated feeding chains. For a fish lineage, this is extraordinary. Intelligence here does not sprout from predation or tool use, but from the subtler demands of navigation, social interaction, and survival through cooperation. At the heart of their feeding strategy lies a masterpiece of hydrodynamic engineering. Along either side of their broad mouths extend cephalic lobes, flexible extensions that unfurl into curved scoops when feeding. These lobes funnel plankton-rich water toward the gill rakers, but their function goes deeper. Mantas employ what engineers call ricochet separation, a method where particles are deflected away from clogging surfaces by manipulating flow. Unlike baleen whales that sieve water directly, mantas keep their gill plates remarkably free of fouling. Water swirls, plankton ricochets into capture channels, and filtered water exits clean. This passive filtration is so efficient that bioengineers have studied manta feeding to inspire next-generation filtration systems for desalination and wastewater treatment. Nature solved the problem of fouling millions of years ago with cephalic lobes and ricochet physics. Feeding is not their only ritualized behavior. At cleaning stations, mantas queue with surprising decorum. Each animal hovers in turn, while cleaner wrasses or butterfly fish remove parasites and inspect wounds. Intruders who break formation are often chased or ignored, suggesting a learned social order. For creatures that spend much of their time solitary in open water, these cleaning stations become hubs of interaction, information, and social reinforcement. Some researchers even speculate that cleaning sites may function as meeting grounds where individuals exchange social knowledge, a reef-based forum for a species otherwise dispersed across pelagic expanses. The reproductive strategy of mantas reflects the opposite of abundance. A female carries a single pup after a gestation lasting over a year. Inside, the embryo initially develops from a yolk sac, then receives nourishment from uterine secretions, sometimes described as uterine milk. When born, the pup is already nearly a meter wide, fully capable of swimming and feeding, but reproduction is staggeringly slow. Females may breed only once every 2 or 3 years. This means population recovery from declines is glacial. For every giant school of mantas sweeping through a channel, each individual represents decades of survival investment, not just a fleeting generation. Their longevity and intelligence make them vulnerable in another way. They remember and return. Mantas exhibit site fidelity, repeatedly visiting the same seamounts, cleaning stations, and feeding slicks year after year. This predictability has long been exploited by fisheries. In parts of the Indo-Pacific, mantas have been targeted for their gill plates, used in traditional medicines with no proven efficacy. A single ray yields plates that fetch high prices, but the removal of just a few adults can destabilize local populations. Unlike fast-breeding fish, mantas cannot replenish quickly. Their genius filter, built for plankton, becomes a fatal weakness against nets and harpoons. Adding to this is the invisible threat of microplastics. The same cephalic lobes and filtration mechanisms that so elegantly separate plankton also trap plastic particles. Studies show manta gill rakers often bear embedded microplastics ingested alongside zooplankton. These plastics, carrying toxins and persistent organic pollutants, accumulate in tissues with unknown long-term effects. A creature whose intelligence evolved to navigate vast pelagic deserts and whose filter perfected physics itself is now being undermined by pollutants no larger than sand grains. Yet the manta's influence on ecosystems is not passive. By grazing plankton in vast sweeps, they regulate densities, preventing blooms that could otherwise collapse into hypoxic zones. By visiting cleaning stations, they support reef fish economies, as cleaner fish rely heavily on the predictable business of giant clients. Even their sheer presence alters behavior. Plankton schools concentrate in response to their feeding, and smaller predators follow to exploit leftovers. Like the green sea turtle in seagrass beds or the lobster in kelp forests, the manta is not just a consumer, but a shaper of systems. What makes them most striking, perhaps, is their charisma. Divers encountering mantas often describe the gaze, the way a manta will circle back, approach, and hover eye to eye. Unlike most fish, mantas do not bolt at the sight of humans. They appear curious, even playful. Whether this represents true curiosity or simply the byproduct of their calm glide is debated. But paired with their large brains and social behavior, it is hard to resist the sense that they occupy a mental world richer than most fishes. In an age where animal sentience is being re-evaluated across taxa, mantas stand at the frontier, proof that intelligence need not be tied to teeth, claws, or tools. It can exist in a filter-feeding giant, running not on predation but on plankton, yet their future balances on a knife edge. Their reproduction is slow, their populations fragmented, their filters vulnerable to both exploitation and pollution. Conservation efforts have begun, with mantas listed under international protections and ecotourism replacing hunting in some regions. In places like the Maldives and parts of Indonesia, manta watching generates sustainable income, offering hope. But enforcement is inconsistent, and illegal trade persists. Without vigilance, the genius filter risks becoming another ghost of the Anthropocene, remembered in studies and photographs, but absent from the currents it once commanded. Back at the reef, the manta at the cleaning station completes its slow roll, wings rippling as it rises into the current. The wrasses scatter, their work complete. With a single downstroke, the manta vanishes into the blue, cephalic lobes unfurling as it glides into plankton-rich water. Its brain, larger than any other fish, guides it toward invisible slicks. Its filters sift life from water, while its very biology absorbs threats it never evolved to face. The manta is not just a giant shadow gliding through the sea. It is proof that intelligence can grow from the act of filtering, that elegance can arise from patience, and that even the largest creatures can live on the smallest of prey. The lesson is sharp. Manta rays are high intelligence running on plankton, and they now glide on a razor's edge between survival and decline.